So we're now going to start talking about the structure and function of DNA. And so we've already discussed DNA prior to this. We know that it's a nucleic acid. We know the different bases, and we know some of the chemical components of it. But we're going to go more into depth about what DNA is actually, how it's structured, not just the bases, but kind of its three-dimensional structure, and then also the function of it. And so just a little bit of history, because the discovery of DNA and the shape of DNA is something that won the Nobel Prize in science. And so it's important to kind of recognize kind of how this came about. So in the late 1800s, it was recognized that DNA was a chemical component of cells. Okay, and so what that just means is that they were able to look at the chemical makeup of cells and start to pull out kind of different types of molecules, and they found, DNA. So being able to recognize the sugars, the phosphates, ribose, deoxyribose, things like that. So they know that DNA is a component of the cells, okay? When we get to the 1950s, a series of experiments are going to take place that are going to then convince the world that DNA is the hereditary material. Okay, so we'll say a series of discoveries had convinced the scientific world that DNA was the molecule that acts as the hereditary material. Okay, and so what I want you to kind of realize is that this comes way after Mendel's work. It's also going to come after Darwin's work. We haven't talked about evolution much yet, but both Darwin and Mendel were working in the late 1860s on. And it wasn't until the 1950s that DNA is actually going to be kind of pinpointed as the hereditary material, okay? So this is a very important step in understanding kind of how DNA is going to start to work, okay? Because we know that we can pass on traits from one generation to the next, but what is the material that actually does that? And so we know it's DNA, but it's going to take some steps to actually get there. So just a little bit about the structure of DNA. This is purely review right now, so hopefully this is very comfortable. So we have DNA and RNA. These are going to be our two nucleic acids. So nucleic acids. Okay, so both of the names, this is deoxyribose nucleic acid, and this is going to be ribonucleic acid. So these are nucleic acids. This is the polymer. Okay, and they're going to be made out of nucleotides, which are the monomer. Okay. And there are 4 nucleotides in DNA. Nucleotides. Those are going to be adenine. We're going to have, thymine, guanine, and cytosine. Okay, and we normally just call these A, T, G, C. Now what I want to mention is that thymine, again, this is going to be a DNA only. It is not going to be found in RNA. So this is going to be DNA over here. And then we're going to talk about the RNA nucleotides. And so basically in RNA you have A, G, and C still, but you're going to have uracil, which is going to take the place of the thymine. So this is only found in RNA. Okay, so in all we have basically 5 nucleotides, but you only find 4 in each of the main types that we're going to talk about. So DNA contains ATGC, RNA is going to contain AUGC. Okay, a reminder, A is always going to bind with T, G always binds with C, so that's going to be in DNA. And so for RNA, we're going to have A always binds with U and G always binds with C. Okay, so again, when you're in RNA, you're always going to just, wherever there was a T, you would then put a U. Okay, and this is going to tag RNA as something different from DNA. Another thing about the DNA and RNA is that there is a sugar phosphate backbone. Okay, and so here is kind of an image of our kind of one strand of DNA. They can also be called a polynucleotide. I don't really use that term very often, but if we just take one of the strands of the DNA, you can see the different bases. Okay, so these are the whole thing here with the blue, the yellow, and the base are going to be considered the nucleotide. And so you can have the 4 different types. And then when we have the actual DNA, they're going to bind to each other with covalent bonds. All right, and then those are then going to bind up with another strand of single DNA, and are going to bind with the corresponding bases. Okay, so A would match with T, C is going to match with G. Okay, so a little bit about the actual structure of DNA, not on the kind of the nucleotide side, but actually this whole entire piece of DNA, which we call the double helix. Okay, so how do we actually know that that is the actual structure of the DNA? So the double helix is one of the very, very important findings that kind of allows us to understand how DNA is structured, and it also determines how it functions, okay? So the discovery of the double helix is kind of given to 2 individuals, although there's kind of a controversial story behind all of this. But we have 2 scientists. We have James Watson and Francis Crick. And I'm going to link a video for you guys to watch that kind of talks about this whole situation. So James Watson and Francis Crick were working and trying to figure out how the DNA was actually structured. Other scientists had already figured out some other things, like there always was the same amount of A's as there are T's, and the same amount of G's as there are C's, so you can tell that A's goes with T's and G's goes with C's. But they didn't really know how it was structured. And it wasn't until they were visiting another scientist and saw an image that was created by a woman named Rosalind Franklin. So I think that Rosalind Franklin is an extremely important character in this kind of story. At the time when all this was going on, she was completely pushed back. But what Rosalind Franklin did is she actually took pictures of DNA. She crystallized DNA, and she took pictures, and she took a picture, and her famous picture is called Photo 51. And so this is what this looks like. This is actual DNA. And it may not look like much to us, but for the individuals that knew how to work with this, what this told them is that you actually have a double helix shape for the DNA. Okay, so the DNA is going to look like this. This is a double helix, so you can see how it kind of twists and swirls. This is a double helix, and this picture, kind of looking down on the DNA from above, shows that structure. So when Francis and James went to visit this scientist, they saw Rosalind Franklin's photo, and they actually stole it. And that photo was kind of the last little thing that they needed in order for them to develop the idea of the double helix. And so they basically stole some of her data that she worked on, and ultimately they got the Nobel Prize in Science and Rosalind Franklin did not. Rosalind Franklin is kind of a sad character. The type of work that she did, she dealt with lots of radioactive materials, and she ultimately died at a very young age, probably I think in her 30s, of cancers caused by her work. But today she is much more recognized as a very important figure in the discovery of DNA and the double helix, and we would not know kind of what the structure is without her. So if you watch the video, I'll kind of give you a little more background about that whole story with Rosalind Franklin. But the main thing just to recognize is that Rosalind Franklin ultimately took the photo that allowed for Watson and Crick to develop their idea of the double helix. Now, the double helix is really important because it then immediately suggests a possible copying mechanism for the genetic material. So let me just write that down. And this is actually something that kind of both Watson and Crick said. So the structure of the double helix immediately, so this is their quote, suggests a possible copying mechanism for the genetic material. Okay, and so what happens is that the structure of the DNA kind of points toward how we're able to have the type of reproduction that we have and the type of inheritance that we have. So we're going to talk about how basically this double helix can be separated, it can be read and copied, and then closed back together and conserved. And so we'll talk about how that takes place in replication. Okay, so the structure of DNA points toward a molecular explanation for life's unique properties of reproduction and inheritance. Okay, so because of the double helix and how the DNA is structured, it can easily be broken, read, and then kind of put back together. Okay, so now we're going to start moving on to kind of more about how the DNA is going to actually be functioning. So a topic that I want to introduce you to, this is called the central dogma of biology, and this is extremely important. I always put a lot of questions about this on the test. Okay, and so what the central dogma of biology, a dogma is going to be a principle set down by an authority and kind of saying that this is true. And so this is going to be a statement that has been put down by biologists saying that this is how the information stored in DNA is going to be converted into protein. Okay, and so this is the central dogma. So you have DNA. DNA is going to be turned into RNA. And RNA is going to be turned into protein. Okay? Now, this is only a small portion of the picture because it doesn't tell us how we actually do this. And so we have to add in some more details. So the process of taking DNA and turning it into RNA is going to be called transcription. Okay? And we'll talk about what transcription how it happens and everything that, is necessary for that to take place. To go from RNA to protein, we're going to do something called translation. Okay, and we'll talk about how that takes place. The whole purpose of this is to take kind of the stored copy of ingredients or directions of how to make a protein and be able to take it to a place where it can actually be made. So DNA is stored in the nucleus. Proteins have to be made in the cytoplasm, okay? So this is stored, so this is in the nucleus. This is in the cytoplasm. Okay, because we're going to talk about ribosomes, which are going to be what actually produce the protein or translate the proteins. DNA cannot leave the nucleus, and the ribosomes can't go into the nucleus. And so what we have to do is we have to make this intermediate structure called RNA, which is going to be able to leave the nucleus and go out into the cytoplasm. Okay, so this RNA is really important because it can leave the nucleus and go to the cytoplasm. All right, and so we'll talk about this we're gonna call this messenger RNA. And so it's basically taking the message of the DNA, the instructions, taking it out to the cytoplasm, okay? Because again, DNA cannot leave. One additional thing I wanna put on here is how do we make more DNA? And this is something we've sort of talked about, but we haven't gone into a lot of detail. But the way to make more DNA is called replication. Okay, and I'm going to start off talking about replication first. Replication also is going to take place in the nucleus. Transcription takes place in the nucleus because anything that's going to deal directly with DNA has to be in the nucleus. Okay, so we're first going to actually talk about replication, talk about how DNA is made, how you make more copies of DNA, and then we'll move on to transcription and translation. All right, so starting with replication. Okay. Okay, and so remember, replication is going to be a process that takes place during interphase of the cell cycle. And what replication does is it takes DNA and it makes more DNA. Okay, so you're just making more DNA. That's the whole process. So what's going to happen is that we're going to start with a set of DNA, and I'm not going to draw it in its double helix form because that's too difficult, but I'm going to draw the two strands. And I'm not going to draw all the bases in, but right here you'd have A, T, A, T, G, R, C, C, G, G, something like that. These would be the bases, and they're all bound to each other based on their complementary binding. So what's going to happen in replication is that we're actually going to split these two strands apart, and we are going to make new DNA. And we're going to use the old strands, which we're going to call the parental strands, as templates for the new DNA. Okay? So what we have we have these template strands, which are going to act as the template to then make the new DNA, okay? And so we're going to call these parental strands. Okay, and then the pink ones are going to be kind of newly synthesized daughter strands. Okay. Now, when DNA replication takes place, we have something going on called semi-conservative replication. Okay, so I'll write that, semi-conservative. Okay, because this is what could happen. So we have our two strands of DNA, and let's just say we go through the process of replication and we make two new strands of DNA, but we keep the old strands together and we put the new strands together. Okay, so that is a possible what could happen. Okay, this would be considered conservative because you're kind of keeping the original strands together. Then our other type of process, which I kind of showed up here, is that you have something called semi-conservative, where you have an old strand with a new strand. Okay, and so this is called semi-conservative. And so a scientist did an experiment where he was kind of marking he marked the kind of the nucleotides that were going to be added, and he realized that the new nucleotides add to these old strands, and they kind of stay in this semi-conservative form. So we basically can cross this out because this does not happen. This semi-conservative is how we have DNA replication. So at the end of DNA replication, you're always going to have an old strand with a new strand. Okay, and so that's going to then allow for kind of the passing of these kind of the genetic material. All right, so when we have replication, I'm going to kind of draw this out in a little bit of a more detailed way. And so we're going to have kind of our two strands. I'm going to draw them a little bit thicker this time. Okay. Okay. And I'm also going to show part of the DNA that's been opened. Okay. And I'll explain how DNA is actually split. Okay. So this part of the DNA has actually been opened, and we're going to start making new DNA right here. Okay. Now, when we are actually looking at the DNA, the way that the nucleotides bind together is in a very specific way, and they bind in a way that makes it so that we have something called a 3' and 5' end. Okay, so the 3' end means that there's a 3' spot open for a new nucleotide to come in to bind, and it has to do with like the carbon, and the 5' end is going to have, a carbon that has the 5' that's available. Okay. What happens is that you always have 5' and 3' running in the opposite direction. So if this strand is 3' to 5', this one has to be 3' to 5'. Okay, so they can't go in, the two strands have to go in opposite directions. Okay, because they're going to be going the different directions. Okay, the strands that we are going to be actually replicating, we give them names. And so one of them we're going to call the leading strand. And the other one we're going to call the lagging strand. I'm going to explain why we do that. Okay. So when we're actually going through the process of DNA replication, when the DNA is actually pulled apart, and we'll talk about what enzyme does that, the leading strand is going to be able to be made kind of in a continuous synthesis from the 5' to 3' direction, okay? Because the new strand we're going to add on is going to be 5' to 3'. Okay, the lagging strand is not going to be able to be made in the same exact way, okay? And it all has to do with this enzyme called DNA polymerase. Okay, so DNA polymerase is an enzyme, and what it does is it's going to kind of synthesize new DNA by joining nucleotides together. Okay, so it's actually going to kind of add on the nucleotides. Okay, and I'm going to show you with this in a second with actual letters, so it'll be a little bit easier to kind of see. Okay, and so the DNA is by joining the nucleotides together, and it only works in a 5' to 3' direction. Okay, so it's this little enzyme that could come in, and it can start adding on new DNA. And actually, let me use a different color for my new DNA. So it's going to start adding on new DNA, but this one, it can only work in this direction, which is going to be the 5' to 3' direction. Okay, so this one is going to be able to be made in a continuous strand. So as this starts to separate, it's going to be able to continue in adding on new bases. Okay, and so just a little thing. So if I was to look at this more carefully up close, and I'm going to make up a random sequence. So Okay, so let's just say we're kind of separating our DNA. Okay, and so this is going to be our leading strand. So our DNA polymerase is going to come in and it'll start adding on complementary bases. So every time there's an A, it's going to add a T. Every time there's a T, it's going to add an A. C, G, T, T, A, T. Okay, so that would be one strand. This other strand, okay, is going to be made, but it has to do it in a kind of interesting way. So DNA, again, you know, it's going to have these different directions. So this DNA is going to be made like this. So this is going to be 5' to 3'. And so then they're going to have to do 5' to 3'. And the reason why it has to do this in non-continuous is because as this opens up, you're going to have to start over again. Because if you just continue on going this direction, all of this DNA is not going to be replicated. And so what's going to happen is that you're going to have to keep on adding on new little strands. These will eventually all be joined, but this has to do it in a non-continuous way. Okay, so this is going to happen non-continuous. All right. Okay, so the other strand, it will be very similar. It's going to be in non-continuous, but you basically, it would make a corresponding strand. And so, you know, you're going to have your matching complementary DNA bases added. Okay, and so at the end, we're going to end up having, you know, a strand of the black with a new synthesized daughter strand. Okay. Whenever you're dealing with replication, remember, you're going to keep your T's still there. There's no U's. You still keep everything the same. Okay, so we're not changing to RNA at this point. We're just making more DNA. Okay, so I wanted to mention just a couple different enzymes that are really important in this process. So I've talked about one. DNA polymerase adds on the DNA nucleotides. Okay, so it's going to add on A, T's, G's, and C's. Okay, you're going to hear about an RNA polymerase. That one will add on a UGC, okay? But if you're talking about DNA polymerase, so let's just write this again. So DNA polymerase, okay, so it's going to add on A, T's, G's, and C's, okay? Another really important enzyme is something called DNA helicase, okay? DNA helicase will kind of make the two strands fall apart. Okay, so this is going to unwind double-stranded DNA. Okay, so when we have the two DNA strands, there's going to be an enzyme like helicase, and the enzyme like helicase is basically making it so that your DNA is no longer touching each other, okay? So that you can come in and start making the new DNA. All right. Another little thing, so I talked a little bit about this lagging strand, and so I want to draw this lagging strand a little bit larger. Okay, and so the lagging strand is going to be this 3' to 5', and the new DNA being made is made in the fragments. Okay, each of those is 5' to 3', and they'll continue on. I need to kind of make it so you can see the, once this opens up more, you'll add on another one, and you can just keep on going. So these little fragments right here are called Okazaki fragments. Okay, and there are little gaps in between. And so what the enzyme that fills in those gaps is going to be called DNA ligase. Okay, so it's going to join the ends of two strands of DNA together to make a continuous strand. So DNA ligase will come in here and will then start connecting these, so then you have a continuous strand. And then as it moves on, you add another one, it's going to continue to do that so that you have a continuous strand. So at the end you have, these full strands. All right. So that is going to be kind of the main thing you're going to need to know about replication. So big things about replication. Okay, you start with DNA, you end with DNA. Okay, so you're not making a new nucleic acid in the sense like a new type. You're just making more of the same stuff. Okay, you are going to be using DNA helicase, DNA polymerase. Okay, remember those are going to add on the bases, and DNA ligase. Okay, there is both a leading strand and lagging strand. Okay, and then DNA replication is semi-conservative. Okay, one other thing I want to make sure that for DNA polymerase only works in a 5' to 3' direction. Okay, and the reason why this is important is if you were doing kind of the lagging strand, if it could move in the other direction from 3' to 5', then it would actually be able to do a continuous strand on this one. But it does not work in both directions. It only will go from 5' to 3' direction. Okay? All right. When replication is actually happening, you actually will have kind of multiple areas of the DNA being replicated until you kind of have all of it kind of separated. New DNA is going to come in and you'll ultimately have the two strands by themselves. And so we call these little places where the DNA replication is getting started origins of replication. Okay, and so there are different parts of the DNA that have little sequences that then say like this is where replication needs to start, and they will continue on until you completely have new DNA being made. So, okay, so DNA will be being made in these sections until ultimately the whole DNA strand has been replicated. All right, so now we're gonna move on to the next step. So this is just how replication works, but we were talking previously about kind of the central dogma and how do we take DNA that has all these directions to, make you look like how you look in all the different proteins and enzymes that need to work in your body. How do we take this and get it to protein? Okay, and so this first step is going to be called transcription. Okay, and so this is kind of the first step of what we would call gene expression, because we're now going to start having parts of the DNA being read and kind of expressing those genes. Okay, so let's see. This is going to be called transcription. If you know anything about what the word transcription means in a non-biological sense, it has to do with kind of maybe transcribing or writing something. So taking one version of some type of written document and transcribing it, so rewriting it in a slightly different way. And so that's what we're going to actually do. So this is going to be taking DNA and making RNA. Okay, and so remember, DNA and RNA are different. DNA has deoxyribose as its sugar. RNA is going to have ribose. DNA has ATGC. RNA is going to have AUGC. And so that transcription is going to slightly change what's happening. Okay? Now, all of this again is going to be called gene expression. So if we have a gene that's important for eye color, we need to express it. We're going to need to read that region of the DNA that has that gene a part of it. okay? So we're gonna use some similar enzymes that we've heard of before. And so one of the enzymes is gonna be called RNA polymerase. Okay, and so in this case, instead of adding on a TGC, the RNA polymerase is going to be adding on a UGC, and it's going to build a single strand of RNA. That's another thing I need to put over here. So this is 2 strands. RNA is gonna be 1 strand, because we only really need 1 copy of the actual gene, okay? So they're gonna have RNA polymerase that's gonna add on the AUGCs, and this is only used for transcription, okay? And we're also gonna use helicase again, because we're gonna need to separate the DNA strands so we can go in there and actually read the DNA. All right, so DNA is going to have different sequences within its actual structure. So different parts of the DNA are going to hold these regulatory sequences that are going to tell kind of the RNA polymerase, this is where you need to start transcribing the DNA, okay? And so we have different kind of parts of transcription that are going to take place. So the first part is going to be called initiation. Okay, so this is going to tell you like the initiation, so the starting of the DNA being transcribed. We're going to have something called elongation. And then we're going to have something called termination. So initiation is going to be when a specific sequence of the DNA is going to be read, okay? And so I can show you this, and I'm gonna kind of do an example of this. But we have this strand of DNA, and it's kind of hard to see on here, but there's this DNA right here is green, and there's a sequence of the DNA, and it's encoded within the ATGCs. It's going to then be able to bind this RNA polymerase, and the RNA polymerase is going to start and come in onto the actual DNA. It's going to bind to the DNA. Then what's going to happen is that the DNA is going to be unwound, and one of the strands is going to start to be read, and a RNA sequence is going to be made that matches kind of, or is complementary to the strand of DNA that it's attached to. Okay, as it moves down the strand of DNA, it's then going to ultimately hit something called the terminator, which is then going to cause the RNA polymerase to fall off. But what you're going to end up with is just a single strand of RNA that's going to match one of the DNA strands, okay? And so I'm going to draw this out for you and show you what it looks like. And I'm going to use actual letters so you can kind of see the, what the bases would look like. This is a very small sequence because otherwise it would take me a really long time to write this. And again, I'm not going to write it in double helix form. So let me just write down this sequence of DNA. CG. Didn't quite make this big enough for the sequence that I wrote. Okay, and so the other corresponding strands, so again, these are both DNA right now. I'm just kind of showing you what happens before. Okay, so we're going to call one of these strands the coding strand, and the other one is going to be the template strand. And sometimes it's also called the non-coding. So what's going to happen is this is the part that we're actually going to attach the RNA polymerase to, and we're going to add on bases that match to this template strand right here. But what I want you to notice is that this is the strand up here, the complementary one, is the one that is going to have the very similar sequence that we're going to have. Okay, and so I'm going to draw this again, but now I'm going to actually separate my two sides of the DNA a little bit more so that we're actually open and ready for transcription to take place. Rewrite my bases. Oops, that's a C, G, T, A, A. So RNA polymerase is going to come in, and just like DNA polymerase, it works in a 5' to 3' direction. Okay, and what it's going to do is it's going to start adding on RNA bases to match this. Okay, so I'm going to start doing this. Okay, so the match for T matches with A. Okay, this is where our new change comes in. A is going to match with U. C still goes with G. T, A, U, C, U, C, G, U, A. Okay, and so what I just wrote now is my RNA strand. Okay, so remember, A binds with U, G binds with C. Okay, you are going to have T's on your DNA strand. T's are still going to bind with A. Okay, T's do not bind with U, so they're going to always bind with A. So then we have the sequence. So this is our RNA strand. So this part would fall, kind of leave, unattach. So this is what we're going to call this pre-mRNA. Okay. After this is done, this will come back together and you'll have your DNA kind of going back to how it was. So it's been opened and now it's going to close again and we have our mRNA strand. Okay. When this RNA strand is done with this first part, it still has a couple things that have to happen to it before it's ready to leave the nucleus. Okay, because if you remember, we have RNA, but it needs to leave the nucleus, but we're going to have to do something called RNA processing. Okay, and for the RNA processing, I'm going to draw my RNA a little bit differently because we don't really need to look at the sequence anymore. So this is all going to be called RNA processing. Okay, and during RNA processing, we're taking this pre-mRNA, which M stands for messenger, and we're going to turn it into full-on messenger RNA. Okay, and so M equals messenger. Okay. So I'm going to draw our our RNA a little bit different. Okay, so I'm not going to really show the bases, the AU, GCs right now. And what I'm doing is I'm drawing kind of these little sequences on the RNA strand, okay? So when you have a part of the RNA that's actually made from the DNA, there are parts of the RNA that we don't actually want, okay? And so these black parts are going to be called introns. And introns need to be removed from the mRNA before it's ready. Okay, so introns need to be removed. Okay, so we do not want the introns. The pink parts, these are going to be called exons. These are the actual parts of the RNA that we want to keep. Okay, and so the way that we go about doing this process of getting rid of the introns is called splicing. Okay, and so what splicing does is it removes introns from the mRNA. Okay, and so after the process of removing those, we will then have our strand of no introns are left, and it's just exons are left, okay? So if we joined all of these parts, we could just say that's 1, 2, 3, 4, and then we have them all together. 1, 2, 3, 4. Okay, so then this is gonna be kind of the actual part of the RNA that we want to keep and will help us make our protein. Now, another thing that's happening kind of with RNA processing is we are going to add a couple things to the actual mRNA. So this is where we remove some stuff, but we're actually going to add on a 5' cap. So we always are going to have a 5' end and a 3' end. Okay, and so on the 5' end, you're going to add a 5' cap. So 5' cap. Okay, and so what the 5' cap is it's going to help protect the actual RNA, and it's also going to tag it as RNA. Okay, and this cap is going to be, it's a modified guanosine. So it's going to be, okay, so it's capped with something called a 7-methylguanosine. So it's kind of, guanine. So that's going to be added to the 5' end. And then on the 3' end, a bunch of adenines are going to be added. Okay, and so what we call this is on the 3' end, we added poly A tail. Okay, and so what this is, it's actually called polyadenylation, but you're just adding a bunch of A's onto the end of the RNA. Okay, the cap in the tail and the removal of introns then makes this a mature messenger RNA that's ready to leave the cell, or the nucleus, sorry. Okay, so that is going to allow for this to exit the nucleus and enter into the cytoplasm so that our next big step can take place. Okay, so mRNA is going to be what can move out and then is going to be what we're going to transcribe or translate. Okay, so a little bit, let me just review that very quickly. Okay, so RNA processing. Okay, so a couple things that have to happen. So you add 5' cap and poly A tail, so this 3' end. Okay, and remove introns. Okay, and this is through splicing. Right, once it's done, that means that the pre-mRNA is now a fully fledged messenger RNA. So this is ready to leave the nucleus. Okay, so that is kind of what transcription is doing. It's producing this RNA strand, it's single-stranded, it's going to be processed, and then it's going to be ready to leave the nucleus. So now we're going to move on to our last major step, which is called translation. So when you think of the word translation, I hope you're thinking about languages, and so how we can take, English and translate it into Spanish or Portuguese or Mandarin. You're basically taking these words and putting them into another format. Okay, and so that is what we're going to be doing now. So when we do translation, we are starting with mRNA and we're going to make protein. Okay, and so how do we actually do this? And so what I want to kind of talk about now is we're gonna start talking about the actual bases and what those bases mean. Okay, so we have, I'm gonna just write out a sequence of DNA, RNA. Let's get this. Okay, and so I can just continue on writing this. This could continue on. And I'm gonna end it with that. So you're gonna have an RNA strand that's going to have a sequence of the bases, okay? And the way that this is going to now be translated is we have to break up this and read it in a very specific way, okay? And so how we do this is we read in triplets, okay? So we're gonna go in here and our words are going to be the 3 letters, okay? So we're gonna take each base in a group of 3, and that is how we are going to now translate it. So we're going to change this from kind of the nucleotide base into our next type of structure, which is going to be an amino acid, okay? And so what we do is we call these, so we're going to read, so let me just say RNA is translated into polypeptides, so we're making proteins, 3 nucleotides at a time, okay? And I just want to remind you, so we have 4 possible nucleotides. We have A, U, G, C. So there are 4 nucleotides. And if we're going to read them in groups of 3, what that means is that how many different combinations of nucleotides can we have? So we have 4 bases. We're going to read them in 3s, okay? 3-letter codes. Sorry, it shouldn't be multiplication. This is actually going to be, wait, I think you go, is it 3 to the 4th? I actually get my math out. So you have 3 letters, yeah, and there are 4 different possible letters. So then you're gonna have 64 different combinations of these letter codes, okay? And if you remember correctly, what are polypeptides made out of? Well, they're made out of amino acids. Okay, and if you remember, we have 20 amino acids. There are 20 amino acids that we have. And those, you know, we don't need to know the names of all of them or anything like that, but there are 20 of them. And so what these are, 3 RNA nucleotides are going to correspond with are the 20 amino acids. Now you're going to notice that there are 64 different combinations of the letters and only 20 amino acids. So what's going to happen is that there is going to be some redundancy built into this system. Okay, and so what I want to point out to you now is this is kind of the universal genetic language where we can take our different bases and read them in groups of 3 in order to then have them correspond to specific amino acids. Okay, so if you've never seen one of these kind of charts before, they can be a little bit confusing initially. But what I want you to recognize is over here we have the first base of the RNA. So you're always going to have on your, what we call and these are going to be called codons. They can also be called triplets. So these are going to be the 3 letters put together. And so you have your 3 letters. So the first base, whatever's going to be in this first column, is going to be this letter right here. So you're going to say, well, maybe our base is A, U, C. Okay. So we would go first base A. So here's A. Okay. And then on this side right here, this is the second base, and our second base is U. So we would go to this one. So we're in this box right here. We're in U. Okay. And then our third base codon is going to be C. And so then we go over here and we say, okay, A, U, C. This one is going to be isoleucine. So what I want you to recognize is that you can actually have 3 different codons, or these 3-letter codes, that's going to code for isoleucine. You have AUU, AUC, AUA. Now what you're going to notice is that the first 2 letters are the same in all of these. It's the 3rd letter that is different, okay? So this final one, this 3rd letter in this codon, can be 3 possible things and still code for the same amino acid. Okay, and this is going to come into play when we start talking about mutations. So most amino acids are going to have multiple codons that are going to code for them. So like leucine, threonine, alanine, valine, serine, arginine, all of these ones have 4 possible amino acid codons that code for the same amino acid, okay? What you're gonna also recognize is there are a couple things that are not what we consider amino acids, okay? So there are 3 codons, UAA, UAG, and UGA. All of those represent stop codons. So these are not amino acids. When you are translating something and you come into contact with a UAA, UAG, or UGA, it means stop here, no more amino acids are going to be added. Okay, and we have one other very special, this one is an amino acid. This is AUG. This is the only amino acid codon for methionine, and this is considered the start codon. So when I start talking about how translation takes place, the machinery that's going to translate these mRNA strands is always going to be looking for AUG, because this is where it knows to start to actually translate. Okay, so this right here is called the universal code. And this is one of the big lines of evidence for evolution, because if you look at any type of bacteria, plant, fungus, any type of living organism, all of them use the same kind of letter code, so the bases that correspond with the same amino acids. Okay, and so this is a universal code, kind of the universal language that is going to kind of hold in that genetic information. Okay, so how do we actually do this whole process of translation? So I've shown you, you read, you always are looking at the 3-letter code, and then you kind of figure out where you are, and that's the amino acid. But how does this process actually take place? Okay. So we have a couple enzymes we need to talk about, and another structure. So we're going to be talking about the ribosome. Okay, so this is going to be how we do this, translates proteins. Okay, but we also need to talk about something called tRNA. Okay, and tRNA stands for transfer RNA. So what this is going to be involved with is this is going to be kind of this, the enzyme or the structure that brings the correct amino acids to the growing polypeptide. Okay, and I'll just kind of show you what this looks like. I think I have a... So this is a tRNA right here. It's kind of a very simplified structure. This is not exactly what it looks like. I don't think I have the actual very complicated version of what it looks like. But basically what tRNA is going to have is it's going to have this portion on its structure that is going to have what we call an anticodon. Okay, and so we're going to have the anticodon. Okay, and so what's going to happen is that if we have kind of our sequence of mRNA, so I'm not writing out all of the different letters, but the anticodon is going to come in here and it's going to correspond with the actual codon. Okay, so this is the codon, the 3-letter code. This is the anticodon, so this is the tRNA. So the tRNA is going to bind to the codon with this anticodon. So it's going to kind of like little puzzle or Lego pieces where they're going to be able to fit together because they have corresponding bases. And so what's going to happen is that there's going to be an amino acid. So we'll just draw the amino acid as if it's a triangle right here. It's going to bring in an amino acid. And so, for example, this amino acid would be methionine. Okay, AUG always stands for methionine. And so if we kind of moved on, we would then have another, let's just say this one's CCU. We would have another tRNA come in, and it would then have an anticodon that would match up to CCU, which would be GGA. And if I want to figure out what that one is, okay, so G, first letter, G, second letter, A. Okay, GGA. So this is going to be glycine. And so the next tRNA would bring in amino acid, or the amino acid glycine in there. Okay? So the tRNA is going to be called transfer RNA, and it brings in the amino acids to the actual structure. Okay, so here is methionine. So they kind of drew it like that. Okay, and so the next kind of step that's going to happen is that we're going to have the ribosome is going to need to attach. Okay, so the ribosome is going to be kind of the structural component that's going to allow for all of this to take place. So the way that this starts is that the start codon, okay, which is methionine right here, so it's AUG, is going to come into contact with the small subunit of the ribosome. Okay, the small subunit of the ribosome is going to then bind to the mRNA strand, and methionine, this tRNA with methionine, is going to come in and is going to start initiating this process. It's going to bind to it because they have corresponding bases. And once this happens, it brings in the large subunit of the ribosome. Okay? And so this is called initiation. Okay, so the first step of translation is going to be initiation. Okay? All right, and this involves kind of the first time we're going to start talking about the ribosome in more depth. So just quickly before we start talking more about initiation, the ribosome is going to be an enzyme. Okay, it's actually very large, and it is also going to be partially made out of what we call rRNA, which is going to stand for ribosomal RNA. Okay, and so the ribosome is going to have two subunits. It has kind of a large subunit and a small subunit. So small and large. And it's going to be the site of protein synthesis. Okay. So back to initiation. Okay. So we're going to have, okay, so let me just start this initiation. Okay, so occurs via start codon, okay, which is AUG for methionine. Okay, it attaches to small subunit of ribosome. Okay, and part of this whole thing is going to be that the mRNA with its tail and cap are going to be very important in kind of having the two parts find each other. Okay, so the mRNA tail and cap are important eukaryotic. So this is going to be different in different groups, but in the eukaryotes, eukaryotic initiation factors. Okay, so that cap and tail are going to help the RNA kind of find the small subunit, and then it's going to bind to that subunit. Okay. Next, after that, tRNA is going to start bringing in different amino acids, okay, to the actual subunit, and you're going to have your mRNA strand is basically going to be running through here, and we're going to start adding on kind of these different bases, or these different amino acids. Okay, so you can have like just show them different shapes. Okay, and so we're going to start making this polypeptide. So I'm going to show you, it's hard for me to draw, so I'll show you this in actual. Okay. So here we have the codon recognition. So we kind of have our mRNA strands. We're going to have kind of a growing polypeptide chain, and as the mRNA strand moves through the ribosome, these different tRNAs are going to come in and start to bring in the correct amino acids, okay? And they're doing that based on what the actual strand of our mRNA has on it. So these tRNAs are just circulating all throughout this area, and if they happen to come in and have this corresponding anticodon to the codon, it will bind and allow for kind of this polypeptide to actually lengthen. Okay, so here we have the new tRNA that's come in here. We're gonna have a binding, so this is gonna happen through hydrolysis, not hydrolysis, dehydration, where a covalent bond is gonna form between these two amino acids, forming that polypeptide bond, okay? After that happens, you're going to kind of continue to have that new polypeptide bond that's gonna form. More tRNAs are gonna come through, and you're gonna ultimately keep on going until you end and get to a stage where there is kind of a stop codon, okay? So once we get to a stop codon right here, then all of this basically falls apart and you have your polypeptide, okay? That polypeptide then needs to be folded, and other modifications can take place before it turns into what we call a mature protein, okay? So here is just kind of an overview of everything that we've talked about. So transcription takes place in the nucleus, okay? So you have your full DNA, double helix, 2 strands. You're gonna have RNA polymerase that's gonna come in here and is going to kind of create this new mRNA strand. There are going to be introns within the mRNA strand, and so those are going to have to be removed. So we have kind of introns removed, a tail and a cap are going to be put onto the mRNA, and then the mRNA is going to be able to leave the actual nucleus, okay? While that's going on, we are going to have, kind of tRNAs that are going to have amino acids added to them. We don't really talk about this stage too much. You don't need to worry about that. But while that's happening off on the side, we then are gonna have our mRNA strand that's gonna come out here. It's going to bind to the ribosome, and the ribosome is then going to have kind of allow for the tRNAs to come in and start to add on the different amino acids so that we can start making this polypeptide chain. Okay, at the very end of this, when we get to a stop codon, which again, the stop codons are going to be UAA, UAG, and UGA. You do not need to memorize these, but just understand what a stop codon is. It will then have everything basically falls apart. The ribosome will dissociate from each other, and you'll have your polypeptide ready to go. Okay? And so this, again, is kind of what's the whole process of how we're going from DNA to protein. Okay? And so it requires production of this intermediate step of mRNA, and then kind of the process of actually turning it into polypeptides. Okay, so important things to remember about this process is that you read the kind of the mRNA in 3 base codons. Okay, the codons code for amino acids, okay, and the amino acids can be found on here. So you have your 3 letters. You can then have all these different amino acids, okay? Now, what I want to talk about next has to do with when something goes wrong, okay? So when we're talking about this, we're assuming everything's going right, there's no mistakes made, but there are going to be times where mutations take place. Okay, and so we're going to talk about mutations right now. So, and that's going to be, so how do we get to a point where we're making maybe these proteins, or let me show you this one. We're making these polypeptides, but maybe what happens if one of these gets changed, okay, and it's no longer the right amino acid. Okay, so how does that actually happen? All right, so these are going to be called mutations. And these are very important. I want to stress that mutations, okay, these are going to be kind of the ultimate source of variation that allows for evolution. Okay, and we're not quite to the point where we're going to talk about how this happens, but you have to have mutations take place in order for evolution to take place. Okay, and so we always think of mutations as being something that's very bad, and they can be horrible, but they're not always bad. Okay, now an interesting thing is that mutations are going to take place during replication. Okay, so remember when we have our central dogma. Okay. So what's going to happen is that replication is going to be where most of the mutations take place. Okay, and so during this process, something can go wrong and a base can be mutated. Okay, what that means is that the DNA that then ultimately is going to be holding the genetic material will have that mutation, and that mutation is then going to cause issues here and here. Okay, maybe not issues, but it's going to have changes. It's going to have cascading effects on both the RNA and the protein. Okay, and so if you change the DNA, you will change the mRNA, which may change the protein. Not always. I'm going to show you some situations where it doesn't. Okay? So we're going to talk about a couple different types of mutations. Okay, this first type is going to be called a point mutation. Okay, and so let's just say that we have our sequence, and we're talking about DNA again, so no U's are here. So I'm just going to draw the corresponding strands. Okay, so let's just say that when we're going through the process of replication, we're making our strands, everything's okay except, let's say this A gets turned into a C. Okay, so we now have a C that's here instead of what it should have been. Okay, so that's going to be fine. Let's just say down here that this is completely fine. Everything is normal, but for some reason when this other one was adding on DNA, it added a different base, okay? And it turned it into a C, okay? So if that turns into a C, so let's just write this down here. So A, T, C, C, G, T, T, C, G. What can happen is that if this is going to be part of, it's within a gene, so a gene is going to be where that instructions for the proteins are. If this is within a gene, then if we're going to make an mRNA from this, okay, so this is our mRNA. Our mRNA is going to be U, A, G, G, C, U, A, G, and this is going to be a C. Okay, so this right here is different from what it would normally be. Okay, so normally this would be, if this was still an A, this should be a U. Okay, so when we're going through and we're going to actually read our codons, this letter right here is now different. Okay, and so we could say, what is AAG? What is the codon amino acid? And then what would our normal one be? So our normal one would have been actually AAU. Okay, so let's see if these are different. So all we did was change one amino acid. So let me get my chart. Okay, so AAG. AA is lysine. Okay, so AAG is lysine. And AAU is asparagine. Okay, and so in this case, I actually did completely change the amino acid. Okay, I did this on accident. I didn't know I was going to do that. But point mutations, if you change just one base, you can ultimately change the amino acid. If you change an amino acid, that protein will not be the same. Okay, and so therefore it most likely will not be able to function in the same way. If you change the structure of the protein, it's not the right structure, it can have problems, it may not work. So this is called a point mutation. Some point mutations are completely silent, okay? They don't do anything, okay? And so we call those silent point mutation. Okay, and this is when you substitute in a different base. So for example, what if I, instead of did this, it would have been, let's see, instead it turned into a T, then this would have been an A. Okay, and so we would have AAA, and if we look at AAA, that would have been lysine, or actually that one I kind of switched it all over again. So I think originally it would have been, I can't remember. But basically, if it doesn't change the amino acid, then it's called a silent point mutation. Okay, so for example, if you change the third letter of, a codon that's going to correspond with leucine, you're basically just going to end up with leucine, because you could change it to a U, a C, an A, or a G, and it's still going to end up being the same thing. Big problems happen when you have a base change that is in one of the first two spots. Because if you change these first two spots, you are definitely going to change what amino acid it is. Okay, and we'll work with this during the lab next week. And so point mutations that happen in the first position or second position of a codon have more dramatic effects than in the third position. This third position is sometimes called a wobble base because it can be multiple different things and still code for the same amino acid. Okay, so those are kind of point mutations where you're just changing a single amino acid or single base that could potentially have an amino acid that's changed. Another type of mutation is something called a frame shift, and these ones are bad. Okay, and these happen when you add or delete, or have an addition or deletion of a base. Okay, and it has to be in some kind of multiple other than 3. Okay, and I'll explain what that means. Okay, so let's just say that we have kind of a normal sequence. I think I'm just going to go straight to RNA. Or no, I'll do DNA. Okay, so let's just say that there has been, when we go through replication, there's an error that happens, and instead of keeping this, what happens is we now, this base gets removed for some reason. Okay, so what this is going to do is that originally, once we got to the RNA, and I'm not going to go out in the RNA, but you would have read the codons like this, okay? But because this has been deleted, what happens is that you're no longer going to read it like this. You're actually going to read it like this. And so it'd be as if you were reading from here to here, And so what this does is it changes your reading frame and shifts it so that it's no longer the same amino acids, okay? Frame shifts are almost always destroy the actual protein. If you remove 3, what happens is because the codons are 3, you'll just remove one whole amino acid, but all the other amino acids should stay the same. But if you do anything other than 3, you'll have a problem. This also would be a problem if you added in an extra one, okay? Because if you add in another or base, you're going to shift how you're reading again, okay? Because you always read in 3s, and so you add in something new or delete it, it's going to cause this to shift, okay? Another type is when you have something called a nonsense mutation, where a stop codon is prematurely put in. Okay, and so if you have, for example, a sequence where there shouldn't have been an amino acid that a stop codon, but there has been a kind of an error, and you now have a stop codon, so the codon turns into one of these groups, what's going to happen is it's going to prematurely stop. And that's going to cause issues because you're not going to get the whole polypeptide chain read correctly. Okay, so that is another potential problem when you're doing those. So those are called kind of nonsense mutations. And that's a premature stop codon. All right. And then a last type of mutation that can happen is something called a repeat mutation. And this is when you basically have a short sequence of bases that just gets inserted multiple times. And so if you have your kind of normal sequence, and then all of a sudden you have this kind of short sequence inserted somewhere in here, and it could be inserted multiple times, up to 10 times it could be inserted. That is also going to destroy the function of your protein. Huntington's disease, which is an inherited disorder, and something called Fragile X syndrome are examples of when these repeat mutations happen. Now, one of the big things to understand is that these mutations have to take place within an actual gene for them to cause issues with the protein that is created. Okay, so if you have these mutations happening somewhere else in the DNA that's not being read, it potentially can cause a problem, but it also could do nothing. Okay. All right.